Regina.
Good morning.
Good morning. How are you?
Good. Quite excited to be here.
Pleasure to see you. So MERA-India is quite a new program here. It's an initiative from ICMR headquarters, and we are just around a 2-year-old program with the mandate of research activity and encourage researchers towards absolutely on malaria elimination. So, I mean, in the first year, we have we received very enthusiastic proposals around the world, and we have funded around 35 studies in various themes like community behavior and engagement and low density infection in the field and migration as well. But this migration theme was a little disturbed because of this pandemic, movement of people here and there.
Right. I've been impressed with the changes in the malaria numbers in India.
Yes. We have funded a few projects on vector biology and control as well. So we have identified some sites, endemic sites of malaria, and on the similar sites we have funded all the themes projects there. So this is ongoing, and we have just introduced the second call of proposal. So MERA-India's secretariat is in National Institute of Malaria Research. I'm sure you have heard about this institution.
Yes, of course.
It's an old institution with a mandate of malaria research.
Just like the CDC.
Yes.
Except they did, yeah. That's why it was created at the very beginning.
Right. We are running this lecture series. Last lecture was recent 2 lectures were one on the COVID-19. It was delivered by young faculty members in Indian Institute of Science, Bangalore. Second was from Dr. Sanjeev Krishna from St. George.
I was impressed by the list of speakers. I know many of them.
I think Dr. Amit Sharma, who is the director of National Institute of Malaria Research, has joined.
Good morning, Dr. Regina. Thank you so much for accepting this invitation from MERA-India. We are really floored and excited about your lecture on Ivermectin. In fact, it's one of the molecules that we are just beginning to look at, and I think one of our papers will be coming out soon on how Ivermectin needs to be studied in Indian context of Indian vectors. I'll send you a preprint of the paper a little later.
Actually, I think it was actually published last week, or at least I got it over the weekend.
Right.
Yeah, from a search that I do on ivermectin, on malaria control. So it was very interesting to read that.
Yeah, so we don't know its effect on Indian vectors, etc. And I think that's where sort of we need to start from scratch. But thank you so much for accepting. I know it's sort of late for you. You're very kind to have accepted this invitation. The time difference makes it very difficult. Of course, it's easier to catch students here during daytime than it is at nighttime. So
right, not a problem. Not a problem.
Yeah.
I would rather be there. I would rather be there, of course, but we do it.
Yes.
We have to do these.
So anyway, thank you again. And Sachin, please take over.
Yeah. So I think we can start now. This is the right time to start. So this is the 8th lecture of the series, and I'm very glad to say that Dr. Regina Rabinovich she agreed to be part of this lecture series long time back. And when I recently contacted her, she was not well, but despite that, she's still recovering, I guess, and she's kind enough to be agreed to this deliver this lecture. So thank you very much, Dr. Regina. And before we start, I would like to introduce Dr. Regina. Dr. Regina has over 25 years of experience in global health across research, public health, and philanthropic sector with the focus of strategy, global health product development, and strategies resulting in impact of endemic population. She's a well-known name in the public health, especially in the malaria elimination program. She holds a medical degree from Southern Illinois University and a Master's of Public Health degree from University of North Carolina. She's the ExxonMobil Malaria Scholar in Residence at Harvard University and the Director of Malaria Elimination Initiative at Barcelona Institute for Global Health. She serves as the chair of the Malaria Eradication Scientific Alliance, hosted by ISGLOBAL, where she led the steering committee of Malaria Eradication Research Agenda. So this lecture is going to be very, very significant and important especially for MERA-India. Dr. Regina spent around 11 years at the National Institute of Allergy and Infectious Diseases at NIH in positioning ranging from epidemiology fellow to the chief of the Clinical and Regulatory Affairs branch division of microbiology and infectious diseases, focusing on the development of evaluation vaccines through a network of US clinical research units. She participated in the Children's Vaccine Initiative as well. She became a founding director of PATH Malaria Vaccine Initiative, which was funded by Bill and Melinda Gates Foundation to advance efforts the development promising malaria vaccine candidates. Prior to joining Harvard, she served as a director of infectious diseases division at Bill and Melinda Gates Foundation, overseeing the development and implementation of strategies for prevention, treatment, and control of infectious diseases, particularly relevant to malaria, pneumonia, diarrhea, and neglected infectious diseases. She is a past president of American Society of Tropical Medicine and Hygiene, and she serves as a board of CMMB and the Saving Vaccine Institute. She serves on the Malaria Vaccine Advisory Committee at WHO, the European Union Malaria Fund Advisory Committee, and the Strategic Technical Advisory Group on Disease Elimination at PAHO. She has received numerous academic and professional awards. In 1993, she received at NIH Merit Award for the contribution to the NIH Vaccine Research Program. In 1995, she was given the NIH Director's Award for skillful and successful advocacy for vaccine research in development of comprehensive national and international program plans. So with this, I know I can talk throughout the day if I continue with the introduction. But in interest of time and looking at we are really excited to hear her. So the floor is yours, Dr. Regina, please.
Thank you for that kind introduction. I'll talk a little bit later maybe about these Altamira cave paintings, but it has to be with the need for data to prove what people believe is not so. And when you're talking about repurposing an old drug for an entirely new way of using it, sometimes you feel like you share in the struggle with the original cave painters. So I'm going to touch a little bit about where we are today with malaria and why there's a sense of urgency at a global level. Introduce the concept of ivermectin for malaria. Talk about a small project that was funded by the Gates Foundation on developing an ivermectin roadmap to bring the community together and particularly explore a little bit the use scenarios that were discussed. Talk about the data that we have right now, which is basically based on modeling and some papers that are beginning to be published, but there are a number of key data gaps. So we'll go over the active trials and the challenges that we face. And then the real question is, what do we do if this works? The global malaria context since 2017, as I'm sure you're all quite aware, was that a global stall was documented with the biggest gap in Africa. It was really a call to action that the house was on fire and that everything that could be built into the 2030 strategic plan for WHO needed to be addressed, including the mapping of biological threats, including resistance, gene deletions for diagnostics, resistance to and impact on drug efficacy, and the emergence of new invasive species. But in addition to the biological threats, were changes in the political context. Financing, which had advances the creation of the Global Fund, had also met a stall. And in some sense, global prioritization given the diverse needs of the communities beyond malaria and the realization that other diseases that we face are also quite important. The bottom line, however, was that there's a need for new tools to reach the global goals. The WHO then reported in December, just last month, the World Malaria Report for 2021. And first of all, you have to take a little time to read it. And in preparing for this, I spent actually quite a bit of time reviewing the assumptions within that. It's extremely well written and thoughtful report as it has been over the past decade. And they've updated their methodology for calculating cases and deaths, recalculating it backward to the year 2000. But the challenge is that it's really several different scenarios. Some countries are really progressing. We have the certification of China and El Salvador as malaria free of transmission. We've gone from 4 countries at less than 10,000 cases to 26 countries at less than 10,000 cases. And most notably, for thinking about Asia, this was something I was not aware of had been so successful. P5X has dropped from 18.5 to 4.5 million cases, indicating that a lot of progress can be made with the tools that we have at hand. I also noted that no country since 2020 that has been certified has gone back to sustained transmission. Now, every country can have importation, but if the systems to track, to identify, and to treat are in place, then it does not become a sustained transmission. For last year, the WHO has estimated 241 million malaria cases and over 500,000 malaria deaths worldwide. And this was an increase with the adjusted figures even for 2020 of about 14 million cases compared to 2019 and about 69,000 more deaths. So clearly there's advances in regions and countries, and there's challenges, and these are particularly in Africa. It is estimated that about 2/3 of these were linked to COVID disruption on prevention and treatment. And although that's disappointing, I must say that about 1.5 years ago, WHO with Pedro, Alonso, and others published a paper warning that if active interventions were not made, we faced a potential for twice the number of deaths and twice the number, and then huge escalation in cases. So this is really the result of a great partnership between WHO, the countries, and all of the funders and actors in the space to try to support the implementation of the traditional tools of BETNEDS treatments and diagnostics. Now, so the challenge is for those countries, particularly in Africa, but not only in Africa, where the stall and actual regression to increased number of cases has been faced, is what is the role for what Dr. William Campbell called an elderly drug? He was one of the 2 inventors of Ivermectin while he was at Merck, and where he shared with the Nobel Prize. And reminds us that it was a long-established veterinary insecticide, originally licensed for about a dozen veterinary applications, which is unusual for a drug to burst on the scene that way, and was first approved for human use for its anti-parasitic activity against onchocerciasis in 1987. So basically for worms. It is today extensively used for veterinary indications, particularly for in the north, where there are the resources to use it routinely for treatment. And but it really goes back to the 1980s, where Merck first noted its anti-insect impact, with multiple studies since then have confirmed consistent and systematic effect on an array of Anopheles species, not on other mosquito species, but on Anopheles. It is used today both for individual patient care and for public health campaigns for oncolymphatic filariasis. Scabies is now coming on, and it's under evaluation for soil-transmitted helminths. And most notably, at a dose of 150 mg per kilo, it is mass drug campaigns that have been distributed to more than 3.5 billion doses for public health goals. So this is the drug that we're exploring. It is the only insecticide that is licensed for veterinary and humans, although there are others that are licensed for are being evaluated for veterinary. And I will make a very short comment at the end of two that have increased performance because of the way they would sustain the implementation should it work. So this is a paper that just represents actually a whole body of work that mosquitoes feeding on ivermectin treated volunteers, here human volunteers, at a dose of 150 to 200 micrograms per kilo in 24 hours die. And the die by 10 days, it is remarkably consistent compared to placebo. And for those that do survive, that become affected here lower in the curve, I don't know if you can see my cursor, they can still suffer effects of decreased flying, decreased feeding behavior, and decreased reproduction. So it can still impact on the potential for affecting residual transmission. Now, there was a whole body of work that took place at WHO. The first was a technical consultation in 2016, in which a number of papers were synthesizing the evidence and the challenges and the questions that needed to be addressed were presented. That report was then reviewed by the WHO Malaria Policy Advisory Committee, now called the group, MPAG. And WHO created this WHO preferred product characteristics for insecticides. And they are now updating it, and there should be a new document available sometime this year. I don't hesitate to say when because it's WHO. But I know that it now is attempting to address what preferred product characteristics would look like for other than the traditional ivermectin. So I think it's also recognizing the innovation that's happening in the field. Around that time, it became very clear that individual funders were very excited about this and were funding individual trials. There was no connection really between them. And this is where the small grant from the Gates Foundation, with the goal to increase alignment between researchers, product developers, and funders on the process for advancing ivermectin as a complementary tool to reduce malaria transmission, the creation of a critical path towards the target product profile in at least 2 different eco-epidemiological settings and then rationale for that, and also to develop the regulatory pathway with greatest probability of success. And that's available online. We have this very cool cover. I do want to comment on what I mean by regulatory pathway, because it's not usually what researchers worry about when they're trying to identify a molecule, develop a molecule into a drug, etc. We would go talk to people in the drug field, and they would say, well, yes, but it's vector control. You need to go talk to IVCC. We go talk to IVCC. They said, well, yes, it's vector control, but it's a drug. You need to go talk to the drug people. And everywhere we went, there was a lack of clarity as to what the process would be, what needed to happen, what data needed to be created, and for what community. And so I'll talk a little bit more about that. But that kind of uncertainty is disheartening to investigators. It makes the challenge of providing the right kind of data for a global and national advice even more challenging. And it makes it quite difficult to engage industry to develop or manufacture at scale the kinds of products that you will need. So it sounds minor, but it can be really important. Now, these road mappers, and it was really interesting, we're all competing. Everyone is coming from different communities. That's how the group was created. Ethicists, veterinarians, people with expertise in cluster randomized trials. We had a kickoff meeting at ASTMH in 2017 and really want to focus 1 minute on these youth scenarios, but different work streams. People collaborated. We fought about things. We argued. There were things we did not understand, and as a whole body of work and publications have come out of that with a final product as a report. But I wanted to spend a little bit of time of the use scenarios because part of the challenge when you're talking about a new intervention and residual transmission and a complementary tool is, where do you use it? Where can you best do the initial trials? How do you prioritize? Do you go to a high transmission setting, a low transmission setting? Is it burden reduction? Is it accelerate to elimination? Is it reducing vectorial capacity? Is the target a mammal called a human? Is the target potentially pigs or livestock that live nearby humans? So all of that needed to be laid out so we could think about not only rationale, target blood source, code delivery options, and rationale for code delivery. So this is worth reading if only for that, and particularly depending on where you're listening to this, what it is your country is, what position your country is, and what challenges you want to overcome. Use scenarios, but I think this makes the case that it's really important to dissect that. Now, the theoretical construct of determinants of efficacy for ivermectin is that there are a number of causes for residual transmission, and that is transmission that happens in spite of vet nets and IRS and whatever other interventions the community is taking. And that is that ivermectin is a short-acting drug. It has a half-life that's well defined, it's less than 24 hours. And so the question is, how do you optimize it for the indication that we're talking about, in which the longer that it is in the bloodstream of that mammalian species, the more available it is to mosquitoes who will bite that person or the livestock, and they will receive adequate supply of drug to be able to die. And it's really a three-dimensional structure, and we struggled with this. I included modeling, arguments, and actually then thinking about what would be acceptable in the field, all coming towards a strategy for evaluation. First, you can use higher doses. You can reduce the metabolism or elimination. So that's one approach to increase the impact. The second is, of course, you can use multiple doses. You can develop slow release alternatives, whether they're stints that get embedded or fancy approaches to actually doing slow release and increasing that half-life. And the other is that you can think at the population level and treat more sources, whether it's figure out how to safely treat children less than 50 kilograms, which is not what the community does and the drug is licensed for. How to safely treat women of fertile age, and how to get other species involved in increasing the blood pool, which can affect then the mosquito. And this actually was a conceptual construct that helped us figure out which of these you could trade off, meanwhile ensuring safety to the individuals. We looked at and asked Dr. Hannah Slater to do some modeling. At first, it began with very simple projections on all age clinical incidents. And on the left here, you have intervention at there are 3 doses that are being given. And you have the intervention of 1 times 400, one tablet at 400 mics per kilo. And another was 3 times 300, so give it for 3 days in a row. And clearly, the one finding from this was what's going to be acceptable and has sufficient safety data that it could be presented without requiring huge studies to create additional safety data, because this is a higher dose than is used for onco. And the second was, what's the trade-off between easy delivery of a single dose and an intervention which would require 3 daily doses, which we do for a number of diseases. Is definitely doable, but it's challenging, particularly in the context of a drug which could have alternate applications. So This was, the left is all age clinical incidence and the right is under 5 clinical incidence. And basically the curves look very similar. It's just the numbers. In Africa, the highest incidence is in children. And so the strategy then became you need to evaluate the endpoint in children within the context of a cluster randomized trial. The field observations, one question we had was, if this works, we kept getting asked by funders and others, if this works, why haven't we seen an impact from the 3.5 billion doses that have been distributed for onco and now triple therapy for LF, et cetera? And part of it is that they use, there's some interesting aspects of those programs. First of all, they're pretty smart. Why try to give this drug or any drug during the rainy season, which is precisely when malaria needs to give it? So whether it is short transmission seasons or whole endemic areas, they go during the drier or the dry season. So that's not going to affect malaria, number one. And number two, this is the Alut paper, but it demonstrated, I'm sorry, the code is for the red and the blue, that there's a short difference in field observations following routine mass drug administration after a campaign had gone through, if one bothered to measure the vector. But the challenge is that this kind of data is not under the current policymaking analysis that WHO does for recommendations. They really have been very clear. They want 2 cluster randomized trials of the same regimen and approach to be done in different countries, not just different vector situations or endemicity, but entirely different countries, to give as the basis for full evaluation. Let me see. All right. Another aspect that was discussed is this One Health approach, in which livestock are treated with ivermectin or potentially other drugs. And the concept is that it leverages the proximity of mammals living close to humans to treat them with an insecticide, basically ivermectin functions like that, injectable or oral or subcutaneous, that will simultaneously improve their health via deworming, improve milk production, improve their resistance to other diseases, and add to the sources of treated blood in the community. Now, there are a number of caveats to this that we needed to think through, and that then ended up being a separate paper, but which I've referenced down below. But the caveats, you needed additional data, epidemiologic impact and the value add for humans, not just the veterinary species. Environmental impact. It turns out that ivermectin is excreted in the feces of animals, and they, of course, are in open ranges or in pens, and that impacts on degradation of dung and would have a potential impact for environmentalists. The other one is the concern about resistance, and a particular concern to the veterinary community that also needs to be addressed. There was also a theoretical potential selection of anthropophagic mosquitoes or their diversion from animals to humans. What you don't ever want to do with intervention is to make things worse. So all these are examples of caveats that need to be addressed, and some of them can be addressed through refuges so that not all animals are treated, etc., but others require additional data. And this is an example of how to think about the role of ivermectin in the human, in the animal, and its impact on the environment, and the kinds of outcomes and operational challenges that get faced as you think about it in a more comprehensive One Health approach. We had to do, for the basis of the funder for the trials, a modeled impact on, and their question was seemingly very simple. They wanted to know, as part of the funding decision, what the impact of mass drug administration with this drug would be 5 years after the end of the trials. And you needed to do some sort of analysis. So we did a simulation, identified 9 African countries with stable political scenario experience in doing mass drug administration for whether either SMC or ONCO, et cetera. This is not the countries deciding. This is entirely a simulation, an outside in, if you will. And the modeling indicated that an assumed efficacy of 20% after 5 years of MDA, you would avert 20 to 50 million cases and 36 to 90,000 deaths. All 9 countries were in Africa. And at 40%, of course, this would be higher. The cost per case verdict would be between $2 and $7, which puts it in the range of a very good buy for public health. Now, reminding you, this is a simulation, so trials and actual data are needed. So that took us to the next state. My slide is missing. Okay, we will go on then. I hope that the other one is. There are a number, and you need to look online for update, but there's a tool called Mesotrack. It's broadly available. You can use Mesotrack to identify what research is going on in Mozambique in malaria. You can look at which are drug trials or vaccine trials. What are the number of projects? Which ones are active? What are the clinical trial sites? Who are the PIs? And this was run a little while ago because I noticed while I was preparing for this that the total funding was 21.7, and it's probably on the closer end of $50 to $60 million so far. But the roadmap, our tiny little grant, was in there. New formulations, we know that there's 2 that are under early development. There are modelers that are working, clinical trials that are being done of various different designs, and then effective ivermectin and a number of different mosquito species. This was identified as a gap whenever you're thinking about how to use it. Okay. So here's the trials. Actually, we lost the table, but now we have the map, so this is fine. The one in Thailand is in the same situation as the one in Mozambique, which was they were both supposed to start last year, and because of COVID and shutdown of travel, etc., all research was stopped at that point. So delayed until targeted for next month. The Burkina Faso trial MDA finished December of 2020. Data analysis and cleaning has been ongoing. Talked to the investigators who've also become really quite involved and busy with COVID. The Gambia massive trial, which was a 2-year study, has presented their main results. In year 1, they had relatively low uptake and identified community engagement as a critical element for cluster randomized trials. But year 2, they had coverage around 80% of the eligible population, and prevalence drops by 70% and incidence by 80%. But the lower vector and documented lower vector density and mosquito mortality reduced even 21 days post-treatment. But the interesting challenge is that they had no effect on their measurement of mosquito parity. So that data needs to be reviewed quite carefully. And then the Guinea-Bissau trial, first year completed, last dose September 2022, with 12 clusters per arm, placebo control, and the data I've been told will be ready in 2023, with a lot of discussion because it's relatively fewer arms than the planned Mozambican trial. So these are, and then the Thailand trial, yes, is starting. That one also is testing ivermectin alone, and that's an important point. What WHO has made very clear is they want to see the difference between standard vector control and the addition of ivermectin without additional combinations with SMC or other combination strategies, which actually also look promising. But to be able to understand the value of ivermectin, they want to see ivermectin with a control. So that leads us to the Bohemia trial, which is the one that I'm involved in. And just as an example, because I could have presented this for all of my colleagues' trials, but We've chosen 2 sites with gradient of transmission, different species and rain patterns, different livestock species and human livestock ratios, different housing and practices. So this is what WHO is looking for, ability to contrast one intervention in very different circumstances. The malaria prevalence in refugee is lower. So that means that the effect of malaria clinical cases actually goes through 10 to 15 years of age. So you have a broader population in which to measure classical malaria compared to MOPEA, which has maybe 47%, but what we're seeing even in the epidemiologic studies done prior to the trial is sustained transmission throughout the year, and with peaks at 75%. So that's what's going on. And it is a collaboration formed by 6 partners, led by ExxonMobil and funded by Unitaid. 4-year program with probably a no-cost extension to complete the trials. But the important part is that it's not just the epidemiologic, what I would call clinical trial efficacy, and what vector people call epidemiological endpoint. It also has a very intense entomology sub-study protocol, a very intense social study, social science aspect to understand how the community understands the ivermectin, different than ivermectin for other indications, how they feel about it, acceptability. There's an environmental sub-study protocol exactly to explore the issues that I raised, and a health economics comparing the 3 arms. The 3 arms, well, this is the outputs that for the grant. It's really an enabling environment, regulatory pathway, evidence, a sustainable business case to make sure that if this works, that there are companies willing to manufacture it at the scale that would be needed. And engagement with stakeholders, including WHO, with broad dissemination of results. Where we are with the design, and WHO has agreed to this given the COVID pressure, is that we maintain 2 different field sites, but this is a 3-arm study. A control, which is an albendazole, offering some anti-worming effect to those clusters. Ivermectin to humans, and ivermectin to humans and livestock to be able to show the difference. Each of the trials in the 2 countries is independently powered, and the data will emerge sequentially, which that's really a good thing. But it is an open label cluster randomized control and parallel arm studies. And the open label is because in the time of COVID, we could not get any company to ensure that they could produce, label, and actually get to us in country albendazole that looked like, or placebo that, and albendazole that looked like ivermectin. So literally that was one of the changes. We're answering the question, does ivermectin MDA to the eligible human population in 3 monthly doses of 400 microgram per kilo at the start of the rainy season result in a relevant reduction? I guess this went backwards. That we've got the individual protocols all going through in-country technical, scientific, and ethics review before going to WHO review. And I'm only going to go through the primary objective, but if there are others that are interested at another point, I can share what the objectives for the other protocols are. Efficacy is infection incidence in children less than 5 years in Mozambique and 5 to 15 years in a second site with lower transmission. And safety is the rate of adverse events and serious adverse events, including the difference between ivermectin and albendazole as the referent. And those are the outcome measures that we will be measuring. Now, I talked about the enabling environment, and let me just spend a couple of minutes because this is why we have a diverse team working on this, but this affects the entire community. And the first is the regulatory pathway. WHO created a new process for WHO bringing together, which is a lot of fun and quite challenging, really smart people in the room. WHO pre-qualification, the Global Malaria Program, the NTD program, which has a lot of interest into what trials are being done, because they use ivermectin for, of course, NTDs. Essential medicines, because they hold the pathway to place it on the essential medicines listing. The Malaria Program Advisory Group, and the WHO Vector Control Advisory Committee. And we present to the VCAG, but all the other groups are in the room teasing apart and presenting questions on the protocol and the design. And they're presented to VCAG for eventually the data will go there for a policy analysis and potential recommendation. Indications are that there was a strong policy recommendation. This would be sufficient for the countries to seek support from the Global Fund, eligible countries. And the most interesting learning for me, and this is the path to troubles, because quite often your assumptions, is that we know this is an old drug and it's been used extensively for onco-NLF. I assume that meant it was licensed. That, it's called grandfathering in, was there, it's accepted as a donation. So actually, regulatory agency in Africa, there's, I believe, only one country that had it as a licensed of a pre-qualified product or stringent regulatory authority product. And most African countries have not done this or are just now going through the process as they're interested in ivermectin for other things. So it's part of that pathway that needs to be built, but had not been built. At the time that the roadmap was done, there were no pre-qualified manufacturers, although WHO had put a request for submissions for pre-qualification. And this is where the market is actually skewed, if you think about it. The vast majority of human ivermectin prior to COVID, prior to the NTDs, was really for donation for NTDs. And so a company would say, well, that's covered by the donating company, which is Merck, and the market is otherwise too small and insecure to really make the investment in doing the studies that you need to be able to present to WHO. And so we've already had discussions with generics manufacturers, and our analysis that supply can expand at generic pricing to meet potential demand for the first 5 years following the end of the project, should the results be positive. The other aspect is policy, and policy isn't just WHO. They make general recommendations, but it's a country process. And the country process is not just attending a meeting, it's briefings, it is presentation of data and answering a number of questions, including what data do I need for my country. And so those kinds of briefings are ongoing, and with the potential of including not only potential products, including insecticides, as a future option in the National Strategic Plan, which can hasten the pathway to funding from the Global Fund. The other, so that's now included in our work plans, but there's still questions remaining. How can the label change? Label can be changed only by the originating company, which in this case for the standard tablet is Merck, and that is what's required to present the change to the listing at WHO. And how will countries regulate this beyond pre-qualification, which they haven't had to do for the donated program? How do you engage quality generics manufacturers with the business case and clarify the regulatory pathway? How can you brief country leadership on ivermectin and potential for inclusion in the time of COVID, which means a lot of Zoom calls? And how do you prepare for translation to scale? What partnership and delivery system will be needed post Bohemia and the other trials that you saw? And how does this get done? So these are an evolving list of the kinds of questions that need to be raised. And this, if the trials are positive. The first is, will efficacy results be consistent across sites? And if it's not, how will the advisory bodies review it? Will safety results be consistent across sites? The third is, how do you best deliver ivermectin for malaria through routine country systems? At the first thought, people were saying, well, you can just work with the NTD program. But the NTD program deals with donated drug, and there's never been an assumption at the volume that we would need for routine delivery of three rounds, not one round. And that for malaria, it needs to be delivered during the rainy season, which is not when that these are necessarily compatible. So this is a challenge that needs to be done. Is it health extension workers? Is it volunteers? Is it faith-based organizations? And there are many countries that have experience with campaigns, and I'm sure some of the solutions will vary from country to country. Financing for the product and for delivery. For delivery has been a challenge for the NTDs, even though they're getting a donated product. And is there a role for ivermectin for malaria for other use scenarios, such as elimination? And that will probably require a very different project design and evaluation component because there's real differences when you have very low rates of disease in a community. What's the risk benefit for the veterinary indication if the data warrants it? And how do we talk with that community? Those discussions have been ongoing. How do we engage the suppliers? We've talked about the label. What's the time of longer duration ivermectin for both humans and for veterinary? And there are at least two products which are quite interesting and will be launching from preclinical to clinical evaluation and will provide us, if the whole thing works, if the system works with ivermectin, with some options for ease of delivery in terms of not doing it monthly, but perhaps every 3 months, et cetera. Do we need further data for ivermectin in pregnancy? And how do we leverage and develop an optimized combination strategy? I'm sure you've seen the paper with RTSS co-delivered with SMC and the impact that really elevates this question of combination strategies for malaria. And there's a lot of excitement. Hannah has done modeling excitement on combination of ivermectin and SMC in a model system is RTSS. And so what does that future malaria intervention look like? So on all things ivermectin, this is my summary of where we are today. I'd like to give thanks to all of the road mappers who not only enthusiastically worked to develop the strategy and put in a lot of time, but we agreed together not to have a lead author, and with the journal, the authorship for this are the road mappers in alphabetical order. And to all of the volunteers and collaborators, investigators, funders, agencies, companies who are earnestly trying to figure out how to derive the best outcome from this investment of these resources. So let me stop there and see if there are questions. I'll turn it back to the co-chairs.
Yes, we have requested our audience to paste their questions in the chat box.
Okay, do you need me to go there? I don't know how to get to the chat.
I'll ask. I read it.
Okay. Okay, oh, here it is. Okay, but I'll let you.
Yes, so somebody is asking, this prophylactic use of ivermectin would not increase drug resistance against other parasites?
Well, it has not done so with the NTD programs. So the frequency is different, but I'm not sure what the basis for doing that would be. There is the theoretical danger that we talked about pushing, depending on how it works, pushing it from veterinary to humans, which we of course want to measure that it doesn't happen. But in terms of affecting other diseases, other infections, and particularly the helminths, the real concern is resistance. Resistance either in humans or in veterinary. And that will just need to be monitored and managed, as resistance always is.
Yeah, and in continuation, what about the toxicity induced by long-term use?
Well, this is not long-term use. It's a very short-acting drug, and that's one of the questions the longer-term drugs will need to deal with. And all evidence in PK modeling is that it is largely gone from the system, but it all leaves the system by the time the next dose is gone. So a month after the third dose, the ivermectin is gone from the human body. Now, there's some interesting work that's going on understanding metabolites of ivermectin. So you need to make sure that you're measuring the relevant molecules. But at least based on the NTD experience, that has not been an issue.
There is half-life of ivermectin is short, right? So when administered in humans, will it be able to impact malaria transmission, especially in the areas of perineal transmission?
Well, that's exactly what the trials are designed to show. It is clear that at the end of 2 weeks or 3 weeks, the mosquito is getting a far lower dose. It's not zero, but a far lower dose at the end of 2 weeks than they do in the first day or in the second day. It drops off. It drops off as goes along, and I showed you the curve. The modeling indicates potential for impact. And I think there are funders who are very committed to the concept that this really should work, but it would work optimally with a longer lasting drug, whether it's a different delivery vehicle or slow release formulation. There are a number of ideas. And I have to say I agree with that. On the other hand, if we can demonstrate impact with the drug that we have at hand, we've advanced, I hope, the progress towards having better products and created incentives also for those better products because of certainty of market.
Okay, so we have here Dr. Manju Rahi. She is a senior scientist at ICMR headquarters, and she is in ECD division, and she is a nodal officer for vector-borne disease program at ICMR headquarters. So may I request Dr. Rahi to please?
Yeah, thank you, Sachin. Thank you. I didn't need that introduction. Thank you, Regina, for such a wonderful and thought-provoking lecture. And I'm deeply interested in ivermectin and vector control in general. And this is, I think, very exciting as a future product. And I think your talk about the multiplicity of and the confusion about the regulatory mechanism, you have actually touched a raw nerve, and that's so true. Where there is an intersection of the intervention strategy, like DEC salt. When you fortify the normal regular salt with DEC drug for lymphatic filariasis, whether it is a salt or a drug, nobody is sure who will regulate. So the door you knock, they turn you away that this is not mine or this is mine. So there is a lot of confusion similarly here. And there are many more examples where there will be a lot of confusion. Even yesterday, I was in fact in a meeting on Wolbachia. So whether it's a vector control or whatever. So that's very true to get clarity for the countries, what regulatory mechanisms we go through. But my specific questions as far as India scenario is concerned would be, India's malaria situation is very, very heterogeneous. And some pockets will be highly endemic and the very high transmission. Maybe ivermectin could be targeted in those areas. But can be like India's major vector is Anopheles sculiciphasis, and that is majorly zoophagic and zoophilic. So will that just targeting cattle and the animals, will that be sufficient to have an epidemiological impact? Maybe that we can see when we do the trials actually. But when the 70% of malaria is driven by a single species, and can we not include humans in the trial and just do the animal trial and see in the beginning, because that would be easier for us?
Yeah, so, I agree that would be an easier design, and we hesitated and didn't do that. And it actually was discussed by the roadmappers because of that theoretical concern that you would push the mosquito population from the whatever veterinary group you would target back to humans. And it's theoretical, it has been raised, and it would be nice to have efficacy data that showed that it worked when given together, and then begin to collect alternative approaches and to gather as much data to address this theoretical concern.
My next and last question, and Sachin would if he permits me, Is there an ethical concern in a country like India where there's a lot of sensitivity about giving drugs, and even for lymphatic filariasis where there is mass drug administration, people are healthy and they refuse to take drugs? Giving a drug to people to control vectors, that too, I mean, they are still susceptible to vector biting and mosquito, transmission of malaria despite taking those drugs, because it's not the way this functions. So how do you convince the community, or what is the ethical angle here? The ethical approvals ecosystem in India may be a little different from other countries, and it's get a little tougher here. So how do you overcome those?
So that's why we have done so much work with community engagement and sharing the challenges and informations across the trial sites. Well, first of all, I think you have to present. It was very interesting. I was in a taxi in Zambia, and he asked me why I was there, and I said, well, I was working on this project, and I described it in 30 seconds. He says, oh, I understand. So you're saying you would be treating all of the people, and you wouldn't treat their malaria, but you would make malaria rates drop. We would have less malaria. And I said, yes, can you please come present that to FDA and WHO? And, he got it precisely. But that's in a highly endemic area, right? At that time, Zambia has actually really improved. So this is the fallacy of prevention. Remember, I'm a vaccine person. I got into ivermectin through a circuitous route. It's about prevention. And when you have a lot of disease, we're all scared to death of COVID. And if they can provide us a vaccine, most rational people would take a vaccine. If there's a treatment that's been shown to work, we want to use it. But when you're talking about the end stage of an eradication program like polio or measles, which in many countries is very well controlled, the risk-benefit perspective changes. And that's a challenge for prevention, especially when you're effective. So in your districts with high rates of transmission, I think that the story can be told, especially if there's data from other places that have demonstrated impact, and you're trying to answer maybe different questions that are a particular relevance to India. But I think you just have to be clear. This will not treat your malaria, but it will decrease the chances that your child or that the pregnant mother will get malaria. And over time, that will make everybody healthier. So, but that's exactly how, I mean, it's not just key opinion leaders or the chiefs, it's actually the community, because informed consent is at every household, right? And this is a GCP trial, which we're doing in the midst of COVID in rural Africa. And I am actually delighted when I see about a 10% decline rate, because that means people are listening and they're voting with their feet, which is actually appropriate informed consent. What has been really interesting is, while they understand and they're hearing from the radio, from community presentations, etc., that many of them have agreed to the census. They have agreed to have the research groups come back. And so I think it's a disease that's really important to them. Elimination, the case for elimination has to be presented. It's a different case, and it has to be presented as a global good, as a good that affects everyone. But that's a different approach to communication, and it's a different set of countries that or districts that need to have that discussion.
I think absolutely we do need to go to an extra mile for that achievement of elimination and eradication. My last point, when you spoke about the reduction 10%, just last question here, I'll not take everybody's time. If I remember correctly, reading someplace, WHO had targeted for insecticides, or for, I don't know, specifically for ivermectin, a drop of 25% or 20% in malaria incidents after the administration for sustained some period of time. So in any trial, has that been achieved?
No, we don't have results yet. There's just not enough. I think that the Gambia results are interesting, but hard to interpret because they co-administered SMC with ivermectin. So that there's a challenge there. And the Burkina trial, I think will be I think, very interesting also to see the results. But we have 45 clusters per arm, and I think they have 10 or 12. So it's just a smaller trial. So this is where, you know, you need to have the power to be able to answer with certainty. And we are powered to answer that question.
Thank you, Regina, for a lovely talk. And then once WHO endorses it, I think it'll be much easier for countries to look up to it and try adopting it. Thank you.
Yes. Thank you.
So now this question is one of your old friends, Dr. Ashwani Kumar from Vector Control Research Center in Pondicherry. Dr. Ashwani Kumar, given you access, could you please ask your question?
Thank you, Dr. Regina. It's so nice to see you after a long time. Thank you. This was a wonderful talk. This had your hallmark. So thank you very much. It was very thought-provoking. And I have a few questions or concerns. One of my questions is the cost related to MDA, because the ivermectin cost is quite high, as you know, per tablet. And it is currently donor-driven. Most of the programs, particularly IDA also, you know, in India against LF, which we have in, you know, MDA exercise every year. And it's hard to procure ivermectin. Challenges are that ivermectin is in short supply many times. And to sustain a very long campaign and in larger geographical areas, this potentially could be a challenge. Even if it is allowed, maybe the companies will ramp up their production and the cost may come down because of that. I'm not very sure if you have some ideas. And another thing is that you're talking about addressing the adverse events or recording them as you go in those trials. There are several of those concerns around, you know, the ivermectin use, although it has a very broad spectrum use against helminthic infections as well. So there are collateral benefits as well, but there are concerns too as regards use of ivermectin. And the other question that I have is that will you propose this, even if it is working as an adjunct strategy to control malaria alongside the other interventions that are ongoing? Or you will be pitching it for use in residual endemic pockets in the last mile of malaria elimination? Sorry for these long questions. Thank you.
Excellent questions. Well, let me start. I'm not pitching it to anybody, but I'm trying to provide the evidence for people to make their own decisions. And there were, I think, we were quite interested as the roadmappers in terms of looking at the elimination scenario, but the sample size and the design that needs to be done for that situation is very different than for highly endemic areas, and where the urgency right now is the places that are stalled. So we've held true to the areas that were identified by WHO, but it is something where it could be evaluated later on with data that shows that it works in areas with a lot of malaria. The issue of procurement, it is a in the U.S. it's, I don't know, catcher in the rye, catch 21. It's a donation program, largely used for humans for mass drug administration. There is relatively little market although it can be used for crested scabies as treatment, etc. So no, they're small producers that can be made. This is not impossible to make. So we took a step back and reached out to the companies that are now pre-qualified, have included generics in the road mapping group, and have reached out to those who are not manufacturing ivermectin. There's great companies in India that are known for high scale, volume, quality product, right? And their answer is, but there's no data. Come back to us with data and we can engage, and we would be happy to potentially play a role. But I can't go back to my board or my boss or whomever because you have no data. So the way to break the cycle of inaction, I think, is to actually bring these trials together. We were fortunate enough to get this funding for a well-powered trial, but to have clarity on the consistency of the data, on what the results are, the cost effectiveness, the population impact, et cetera. The actual AI manufacturers, 8 of 9, there's 9 now, 1 in Spain, but 8 of them are in China. And they won't really talk to groups like us, they'll talk to the companies. So I've talked to the companies to talk to the AI supplier. And it seems like they will manufacture more if there are more requests. They need to have, and so they responded. And I think what we're seeing right now is that it's false, the price is falsely high because it's being used by many for COVID, even though that's a contentious, and we're really waiting for trials to come back to us. It is being used in a number of places for COVID. And I don't know about the quality of those products. So I haven't looked at that, but that I'm sure is causing another, not only the donation program, but the COVID interest is causing another distortion in supply and in pricing. I'm pretty comfortable that should malaria work, new companies, that malaria endpoint work, new companies will come in, and we'll have to figure out, how do you keep enough volume that makes it an incentive for a small number of companies to come in, but to really create it at scale. It's beyond 5 years, where we think it would be taken up, I think you would need additional manufacturers of active ingredient. That's a challenge because this is a fermentation process. And it's largely that skill set and how they tweak it and make it cost effective is largely contained within China right now. And we've learned from the pandemic that you can't have supply just coming from one area. That can create a problem. But I think these issues are solvable. I really do. You asked about safety, and of course, any drug has a safety profile. Any drug, even this one, which targets the biochemistry is such that it targets the parasites and not the human, can in some situations cross the blood-brain barrier. Malaria already creates challenges there, and safety has to be studied. And we were fortunate, and part of the rationale for presenting the 400 microgram per kilo to WHO is that there were enough studies published and a synthesis that was done that 72,000 people had received that dose with very good safety record. So out of the studies that are being done, there will be additional safety data for analysis, and those databases will be made available and published and accessible. There are contraindications. There's some drugs, some HIV drugs that are contraindication, and there's the pregnancy contraindication. So there are contraindications that the programs need to be implemented responsibly. But I also think that can be managed.
Thank you so much. This is wonderful. Thank you once again for your brilliant lecture. Thank you.
Thank you. I see a question in the chat. Ivermectin is used in COVID-19 control.
Yeah, I was about to come here. There are a few questions on COVID-19 related to ivermectin. The question is, ivermectin used in COVID-19 control, can it harm MDA program?
Yes. So you're saying, can it harm the malaria program? Well, I think I think it has the potential for raising a lot of confusion. Um, if you're lucky, your patients are actually telling you what they actually got. And I'm hoping, because of a COVID project I'm doing that has nothing to do with ivermectin, that none of the new drugs have a contraindication with ivermectin, because I'm not sure patients would tell you if they're self-medicating, et cetera. But that's always a concern, and that's always something a physician needs to deal with. Um, the so there's confusion. There's the potential for confusion in conducting the trials. At some point, we will get beyond the pandemic, and then it'll be less of an issue. But right now, I know that it's being used where they can access it in many countries, and it will take another 2 years to have all these trials finish up. And I really pray that the pandemic will be gone by then. So I'm hoping that it'll be less of an issue. I saw a question about 600 microgram has been used for treating COVID patients in Argentina. Yes, we've seen very high doses being used, and actually higher than that also. The original study in Australia, the in vitro study, was at many-fold what could be given to a human. And the in vitro study was sort of pointed to ivermectin or one of its metabolites as having a role, but that dose could not be replicated. So people are trying to push the dose, and I think short term, I guess the ethical bodies have agreed that it's ethical to test it and to get results. I think we were very pragmatic for malaria. Because 400 is in the label in the EU, it's already an accepted dose for humans. And because there were 72,000 people that had received it, we had an extensive, accessible database to provide to the regulatory authorities. So that made it feasible to give us a single dose. I think what you wouldn't want to do for malaria is push the dose so high that you create toxicity that you'd have to back off on. So we built on rather than going as high as that. But that's why I showed you that cube. It's so you really want to add days, which adds delivery complexity, add doses, etc. And you have to look at what you're trying to achieve in your trial in the context and timing that your trial will take place, and that moves that agenda forward with management of risk to subjects and risk to the program. And that's how we came upon 400 after much discussion with our colleagues. There was a question about complementary. I'm sorry that I didn't answer. Right now, one of the hardest things in malaria is to stop doing something. And once things get started, unless you run out of money, it's how do you stop doing something? Right now, because it's perceived as being decreasing residual transmission, it's being done on top of bed nets. IRS has dropped to less than half of what was being done in Africa, so it's less of an issue. But it's being done as a complementary vector tool. And in reality, managing the vector will always be a number of things that people can afford to do and programs can implement. I don't know at what point, and depending on how robust the data are, that people would be willing to stop doing something to do something else. That will take probably a lot more data. It's a really good question.
Another COVID-related thing is there any report on observation on malaria transmission in the areas used ivermectin for COVID-19, and those did not use it?
Right. No, I haven't seen that. I've seen, well, there's a lot of ecologic studies that attempt to show where ivermectin has done that the people have done better with COVID. So for the COVID endpoint, actually, I think in places in Africa, I haven't seen that kind of analysis, and it would be very challenging to capture that data right now. Actually, I mean, right now, precisely those hardest hit countries are the countries in which malaria has gotten worse, but that's a health systems issue. Malaria has gotten worse because of supply chain challenges, because 30% of BedNet campaigns were not delivered, because there's stockouts more than there were before for treatment, because the parents don't want to go to the hospital because there's COVID patients there. All of that enters into a health system disarray. And it's actually not just Africa, it's everywhere. Where patients can't get in the door because the hospitals are for COVID patients, and because they don't want to go to the hospital, so their heart attack or whatever acute condition they have doesn't get treated. This is, I think, a multi-country issue. It's not a rich or poor country. That's how the impact of the pandemic has been.
Okay, so with your permission, I'll take this last question, and because I know it's late night for you. So this ivermectin residues in cattle dung, does it impair the ecosystem from a conservation perspective, especially in agriculture such as nutrient cycling, primary productivity, etc.?
What we believe it has the potential for doing is to prevent dung degradation by killing these beetles that are part of the ecosystem. What we've discussed with the experts who are environmental and veterinarians is there's ways of managing that. There are refugia. There are cows that are being milked, or pigs that are being prepared for market. They wouldn't. There has to be a withdrawal period. So there are practices that are implemented that would decrease this constant pressure. And remember, this is not constant pressure. This is during the peak of the malaria season. So the fact that the other 9 months they're not exposed to ivermectin would decrease the pressure on creating those kinds of problems in the environment. But we need data. And will it be easier in Africa with huge rainstorms? Will it enter into water supply? Will it be just dissipated? Those are questions that need to be answered, and that's why that's one of the protocols. We literally have cows that will be marked with someone chasing and collecting dung from that cow and marking it for analysis. That's the kind of work that needs to be done by valiant research pre-docs as part of their work. But you need to actually get in there and measure it.
So this is promised. This would be the last question. What's your take on the recent reports from Gambia and Aedes species resistance to ivermectin?
So I'm not sure resistance to what. Is it resistance to malaria or resistance to helminths? So I'm not sure I've seen those reports out of the Gambia.
Okay. So with this, I stop here, and I'm sure I'll send you some of the questions here if you.
Okay, thank you. Happy.
And
yeah,
thank you so much for your time. I know it's late night for you, and we are really grateful and thankful that you accepted our invitation for this talk. And this was an appropriate talk for MERA-India as well, and for NMR as well. So
I've really enjoyed it. So thank you very much for the invitation.
On behalf of entire team from MERA-India and NMR, I really thank you, and I thanks to the audience as well for being patient throughout this conversation. And with this, I would again announce that the second call of proposal for MERA-India is open. I request for the audience to please circulate this to your colleagues and institutions, and please apply. And the next lecture would be very, very interesting from Dr. Kamini Mendes next month on 7th. So I will circulate the information. My team will send the information and link and all. So hoping to see you again on during the community Mendes lecture. Thank you so much, Regina. Thank you very much.
Thank you.
Okay,
Bye.
bye-bye.